Good afternoon, everyone, and a warm welcome to this week's Journal Club. I'm delighted to say that Dr. Phil Norville will be presenting today a real-time investigation of a Burkholderia bacteremia outbreak in a Vietnamese intensive care unit. You'll notice there I didn't pronounce the second name of the Burkholderia. I'm going to leave that to Phil because I'm sure he'll do a much better job of it than I would. Can we just advance the slide, Phil? So, welcome to Journal Club. If it happens to be your first time, this is a regular event. We try to give a bit of insight into a topical paper published in the field of IPC. We're really keen that this generates discussion. It's really all about the questions that come in and the productive discussion that we can have. So please do use the Q&A function to get your questions in as Phil is presenting the paper, and that will give us time to go through them and decide which ones we're going to ask at the end. To provide some CPD opportunities to us and to everybody else, we're keen to exercise critical thinking skills. I think it's as much about what's not written in the paper sometimes as it is about what is written in there. We're keen to build confidence so that you can go away from these events and think, yeah, actually, I want to look at a paper and I feel like I have more skills to do that now. Keen to have a network of enthusiasts, and the people here today are exactly that, and ideally have a little bit of fun as well. Just to remind you, all of these events are recorded and made available online after the event. So Phil, how's things with you today? All okay?
Yeah, very good. A sunny afternoon after a bit of rain, which I haven't seen for a while. You've kind of stolen my thunder because I was going to ask you how you pronounce it, but you did
Oh
that in
no,
the
well I
intro.
have no idea. I'm leaving that to you, to your expertise.
Yeah,
So did the poll work or
no,
not? No,
no.
okay. Well, no matter, because I did one in advance of the call for the same reason. So I put a quick poll out on Twitter, sorry, X, yesterday. And I asked the question, how many people have managed an outbreak of Burkholderia? And around 2/3 of people who responded to that poll had not managed an outbreak of Burkholderia, and 1/3 had. Um, I was a little bit surprised that even 1/3 had. They're not that common. I suppose part of it depends on who was answering the poll and how long they've been working in IPC, because I guess you work in IPC long enough, you're bound to come across an outbreak. But not a massively common organism, which is why I think today's Journal Club will be really interesting. So without further ado, I'll hand over to Phil.
Thanks, John. So I'm going to start with a little bit of a different approach today, just because this is an organism that we don't typically talk about in infection prevention control, as John alluded to. And it's also a bit of a tricky one to pronounce as well. So I've broken it down there. I took inspiration from John's Candidozyma advice on how to pronounce it. So it's Burkholderia. It's a gram-negative bacterium, and we don't tend to see it within healthcare or causing infections within healthy individuals, but it's very common in the soil. The people you do tend to see it in are kind of late-stage cystic fibrosis patients. So cystic fibrosis is where you have a defect in the lung, and patients are really prone to bacterial chest infection. And what you tend to see in cystic fibrosis is that the epidemiology in the lung changes as the patients get older. And people who pick up Burkholderia infections in late kind of stage cystic fibrosis, where they've got a lot of progression of the disease, it can often be fatal. So it's a big pathogen for concern there. One of the reasons I've always been quite interested in this organism is that they've got big genomes. So they've got lots of DNA, which encodes lots of different functions, things like resistance to antibiotics and preservatives. But they've also got lots of mobile genetic elements as well, things like plasmids and transposons that can jump around different bacterial genomes and can be transferred by mobile genetic elements. They're very metabolically diverse, and I've got a slide which kind of covers that in the next one. But where we do tend to see them in healthcare, and where they do tend to cause problems in infection control, is when they cause outbreaks, most predominantly originating from contaminated products. I'm going to touch upon this in a little bit of slides time. We have the Burkholderia cenocepacia complex, which is a big group of organisms within the genus, which accounts for around about 24 very, very closely related species. And this is one of the reasons why I think this paper and whole genome sequencing is really interesting, because it talks about being able to have a definition or a definitive answer as to what the species is, given that some of them are so closely related within this BCC complex. So you can just see here some of the features that you associate with Burkholderia and talking about that diversity. So they can do things like contaminate products, both in sterile and non-sterile pharmaceuticals and personal products. They cause pathogenicity in plants as well. So the disease onion rot is caused by Burkholderia. They also are very good at producing some antimicrobial compounds as well. And I was involved in some research a long time ago now, talking about trying to synthesize antibiotics from Burkholderia species, because they use those kind of antimicrobials to outcompete other organisms in a really competitive environment like the soil. And they're also kind of used in bioremediation as well. So degradation of man-made plant pollutants. So you can just see about how metabolically diverse these organisms are, mainly because of their large genome, giving them lots of the necessary tools to be able to work and live in these environments. So before I get into the paper, just really briefly on the kind of IPC implications. If you type Burkholderia outbreak into PubMed or Web of Science, you'll see that there are lots of outbreaks are reported. And they've been reported from mainly contaminated products. And I've just written a list there of the ones that I saw in my literature search. And this is all down to their propensity to be able to survive in the presence of preservatives. So in a lot of those products you'll see there will be lots of additives added to them to ensure that organisms like Burkholderia or Pseudomonas that thrive in wet environments are inhibited. But Burkholderia are very good at withstanding them because they carry a lot of these kind of resistance genes. The majority of times that contamination is identified in these products, it tends to be through contaminated water in the manufacturing process. And this often leads to product recalls as well. And there's a link there to the UKHSI guidance, which is all around the safe care of ultrasound gel, which is one of the big things in the UK, certainly, where we've had outbreaks or cases of Burkholderia reported. So into the paper, which was published in JHI really recently. I chose this paper because I worked on this organism doing a postdoc at Cardiff University some time ago with a really inspirational professor, Professor Eshwar Mahantharalingam from Sri Lanka. And Esh was really, really interested in Burkholderia, mainly from the cystic fibrosis element. But as he did a little bit more work and his research group developed, he showed that they were able to produce certain antibiotics, and they actually synthesized a novel kind of antimicrobial compound from Burkholderia ambifaria species. So I was really interested in the organism ever since then. The other reasons I was interested is because, like we've already said, it's very rare that we see these outbreaks of these organisms. I wanted to know if this was down to another contaminated medical product. I was also really interested to see what the IPC interventions were, and as well this idea of whole genome sequencing. There seems to be a lot of research and energy put into whole genome sequencing now. So I wanted to see how the authors had used whole genome sequencing to investigate this outbreak and, what that led them to decide. So in terms of the outbreak that was being investigated, it was an outbreak affecting 19 patients in a intensive care unit in Vietnam between May and August. To investigate this outbreak, the authors did environmental sampling of high-touch surfaces and some medical devices. And then they were also then performing whole genome sequencing on all available Burkholderia cenocepacia complex isolates using the Illumina platform. And they performed that on 20 different isolates and then some environmental isolates, all of which were isolated from used syringes. The reason why they wanted to look further into this organism and this outbreak was because it was causing big problems in the terms of bacteraemia in intensive care in vulnerable patients, but also that the results that they'd had back previously from the lab were saying that the Burkholderia was coming from a blood culture contaminant. Because this organism is so rarely detected, and we tend to see outbreaks so infrequently, the initial hypothesis was that this organism was being or coming from contaminated blood culture. So they did their environmental sampling, and they were able to identify 3 venous Burkholderia cenocepacia isolates, all from separate syringes, which had been used to deliver IV fluids. They did also sample samples of personal care products and things like hand sanitizers and ultrasound gel, and they found nothing. So they're able to kind of rule out that as a role of the outbreak or the source of the outbreak, which I thought was surprising to me. And this is where, you know, the beauty of whole genome sequencing came in, is that they were able to confirm that all their isolates from both the clinical and the environmental samples were very closely related. They related to or were part of the same sequence type. So very high levels of genetic similarity. They then also were able to look even further using the power of whole genome sequencing to see that the organisms had an array of antimicrobial resistance genes that conferred resistance to lots of different antibiotics that are listed there. However, they weren't actually able to identify the source of the outbreak, but they did identify some limitations in the preparation and administration of IV fluids. So if you look back to my first bullet point talking about the Burkholderia cenocepacia being isolated from syringes, this is where they were thinking that the organisms were being introduced into the bloodstream of those patients. So, you know, a really interesting way of analysing and looking to see the organism being detected in the environment and in those clinical isolates. This might be a little bit small, and it's a little bit of detailed information here, but this is kind of some of the output that you will get from whole genome sequencing when you're comparing the different sequence types. And you can then look at the kind of similarities between them. So the 3 that are highlighted in red there are the syringe pumps, and there's really close or similarity in their sequence types, which enabled them to connect essentially all of those organisms together, both in the syringe, in the environmental samples, and the clinical isolates that had been derived from patients' blood. So they're able to link essentially all of the organisms as the same species and sequence type by doing the whole genome sequencing. So a really nice, straightforward study using whole genome sequencing for outbreak management. What does this mean for IPC? Well, I feel like we're constantly being reminded about the benefits of whole genome sequencing and how it can be used for things like outbreak detection and reducing the transmission of infections. And this just shows that it can be implemented successfully. Also can be implemented successfully in a low and medium income country like Vietnam. But what's really interesting here is that they've used whole genome sequencing to identify complex bacteria like Burkholderia, where often a lot of the traditional methods have very low sensitivity, whereas whole genome sequencing gives you, because you're looking at such a level, you're able to identify the similarities between the organisms and support outbreak management that way. The other thing to point out is that, you know, this requires strong collaboration between different teams within the hospitals, and any organisation thinking about implementing whole genome sequencing, it has to be a combination between a lot of these different stakeholders, such as IPC, microbiologists, and the molecular biologists as well. Again, kind of going back to my first point about that species level identification, really, really important, because the authors reported that a lot of the time, those patients that they were treating had a co-infection, so they're being perhaps treated for something else. And because they haven't got the level of detail and the level of information that whole genome sequencing provides, they might be, you know, treating with the wrong antibiotic. So they might just be targeting, you know, the E. coli infection that they are also treating the infection that the patient with. But what the whole genome sequencing says is that it allows you to identify the organism causing the infection and co-infection potentially, and then provide appropriate antimicrobial therapy. So I really like this idea of kind of tailoring the approach for the individual. And I think when you've got something as complex as Burkholderia in a really high risk setting like intensive care with vulnerable patients and a bacteraemia, then something like whole genome sequencing can really come to the fore. And again, it also highlighted limitations in practice. So we talked on the previous slide around handling and administration of intravenous medications, and this led to some improvements in their techniques and policies around that. So I think, a really strong paper, lots of benefits for whole genome sequencing, and lots of implications for infection prevention control. And that is me. I will pass back to Jon.
Okay, thanks, Phil. I promise I was listening, but I've also managed to get the poll working. So that should be popping up now on the screen. Can you see that?
I can, and I've just voted.
All right, I'm voting now as well. So let's see what comes back in.
And how are you voting, Jon?
Well, I said no, actually, I haven't. I've done like a million outbreaks, but never a Burkholderia one. So I'm in the no bracket. So I think that's probably one of the reasons why this was so interesting to me as a paper, and wanted to read and to look at your blog, which was great, and just to hear a little bit about the organism. So I feel comforted that I'm in the 95% of people who've responded so far who are in the no bracket, which makes me think that X, I don't know, maybe people were making it up.
Have you ever had a case rather than an outbreak?
Yeah. I mean, it's not uncommon to have a Burkholderia case come through, which isn't usually a massive cause of concern, and may, even if it's a blood culture, like you say, may be dismissed as a contaminant potentially. So overall, around about 10% of the audience have managed an outbreak. 90% haven't. It would be lovely to hear from those 10% of the audience who have managed an outbreak, any experience they care to share in the chat or the Q&A. Okay, so whilst I'll leave that poll open in case people want to respond. Thanks for taking the time to do that. So while we're waiting for the questions to come in, just a bit take us back to the background of Burkholderia in general. You showed a slide at the start talking about plants and animals and people. Where does this organism come from? What's its ecological niche?
I think it's the soil. So tend to be, ubiquitous in soil with other things like bacillus, etc. And I think the organism has evolved over time to kind of develop this. What's the right word? Well, it's carved out its own niche within soil, to be able to kind of make sure that it can produce antimicrobials that are going to inhibit the growth of in a really competitive environment like the soil, where you've got obviously lots of different bacteria and organisms. And then I guess just evolved over time to be able to make sure that it can utilize lots of different carbon sources. And then it's then kind of expanded, I guess, outside of the soil, to go and grow on things like, I think it's been isolated from contaminated oil and fuel sources as well. So it's really diverse, but soil is its origin.
Okay, and if we were to sample each other, what are the chances of us being colonized? Is it an organism that you might just find in or on you, and if so, where would it be in a human?
I would think it'd be very rare to have somebody who was colonized with Burkholderia. I don't know if you've got an alternative view.
No, I haven't. I'm throwing it out there. So if anyone in the audience has a good answer to that, that'd be great. So you mentioned the large genome of this organism. How is that significant, and why is that a factor? Does that mean anything in terms of the way that it behaves?
The large genome is one of the reasons why it's so kind of metabolically diverse. So it's got all the different genes which allow it to break down different carbon sources, which means that it can then go and, evolve and develop into different ecological niches. And one of the reasons why it's so widespread as a contaminant, because it's just got this capacity to break down and utilize different carbon sources. So the kind of metabolic diversity is one reason, or one advantage. The other thing which I touched upon was this high number of different genetic elements, things like plasmids and transposons, again, providing lots of advantages around reduced susceptibility to preservatives and antimicrobials. And I think when I was learning about the organism back in my undergraduate days, the large genome essentially was all around the kind of diversity of the organism in terms of it being available or having the opportunity to kind of evolve and work in lots of different niches and environments. And I think this is one of the reasons why we see it in contaminated medical products, because it's just got this ability to survive in environments that organisms, might usually not be able to survive.
Okay, yeah, that makes sense. So, question from Ijoma. Interesting paper. Thanks for presenting it so well. There you go, Phil. Hats off. Good job. Like you said, use of whole genome sequencing has been shown to be helpful in outbreaks to rule in or out cases. For IPC implications, I wonder how we could implement that in real-time practice, especially with the difficulty of the bioinformatics involved.
That's a great question. And I think that's probably the gap that I have personally in my knowledge about whole genome sequencing, is trying to understand how that fits into the real world and real time, and how have you got the right resource, have you got the right infrastructure set up to ensure that if we can generate this information, how can we effectively analyze it? Because we're not going to be doing that as we need bioinformaticians and your molecular biologists, which we talked about within the paper. So I think that's where my knowledge stops in terms of thinking about how we go from a concept to actually being implemented in a real world setting. But I'd be really interested to hear your views on that as well, Jon.
Yeah, I mean, we had Alex Sunderman, didn't we, on Journal Club
We
last
did.
year, and he was describing a process in the States whereby he's pretty much managed to crack it, in that there's a system of proactive whole genome sequencing for selected organisms, and they churn through them. They have the results there, so that as and when an outbreak develops, they can almost see it developing in real time. Now, interestingly, both the question from Ijoma and also the title of this paper had real time in. This was talking about real time investigation. What do you think that meant in the context of this paper? What was real time about it?
I think actually that's probably one of the limitations which I didn't touch upon, but I don't think it is real time. I think they probably used the wrong terminology then, because I think they already had the outbreak before they started implementing whole genome sequencing. And I think as the kind of question feeds into, and what your response was around Alex's work, is that you've got to be doing it proactively for it to be real time. Because a lot of the time, if you've already identified the outbreak, you know that you've got an outbreak. Whereas I think what whole genome sequencing has the power to do is detect those outbreaks to detect those outbreaks in the first instance.
Yeah, so I think I'd probably classify this as reactive whole genome sequencing rather than proactive. But I was just wondering whether there was anything else unusually real time about the investigation.
No.
Okay, I didn't pick up anything either. Interesting. Okay, let's think about the clinical picture presented here then. So I think you said there were 19 patients involved. Did all of those 19 have bloodstream infections?
That was my understanding from the paper.
Okay. And they mentioned that they were concerned that some of these were contaminants and not real infections. Did they go into any detail on that? As in, how many did they consider to be contaminants? What were the definition of a contaminant versus a true infection in the context of this organism and this outbreak?
From what I read from the paper and what I understood was that they initially picked up 3 positives from clinical samples taken from the patients, and they came back as positive for the organism. And they kind of dismissed it in the first instance. So they put it down to blood culture contamination, because such a rare organism and something that you wouldn't normally expect to see 3 cases in whatever period it was that they were looking at initially. They obviously then started to think maybe there is something wider here, sicker patients maybe, or worsening prognosis for them, which then led them to go and do the further investigation. So I'm not entirely clear about where the whole genome sequencing fitted in that kind of duration of the outbreak. But it all started initially from the blood culture contaminants in 3 clinical samples.
Okay, interesting. So now I've got the ability to run polls. I'm just going to go crazy, push the boat out, and I've put another one in there. So I'd be interested to see what people think as we kind of chat away in the background. Did they include anything about outcomes or treatment of these individuals? Were they treated? Did they survive? Did they recover? Or was that not included in the paper?
It was included in the paper, and I think the survival rate was actually quite good.
Okay.
I think that, again, one of the benefits they've highlighted here is that they were able to get real detail on the susceptibility of those organisms to antibiotics. So then they could kind of have that almost tailored approach to the patients. I mean, the other thing as well, I guess, with whole genome sequencing is that if you're identifying the same organism with the same sequence type in all of your patients, the same antibiotic is probably going to work across all of them because the antimicrobial susceptibility profile is going to be the same. But yeah, I think that the mortality rate was around about 10 or 15%. So the majority made a recovery.
Okay, really, really interesting question actually coming from Lisa here. Curious if it was specified whether it was the micro lab or antimicrobial stewardship or infection prevention control team who first suspected this of being an outbreak. And there's a follow-on question there. So maybe they didn't kind of go into that much detail, but was it clear from the paper who initially raised the flag and said we got a problem here?
It wasn't actually. It could be there in the detail, but it wasn't something I picked up while I read it and wrote the slides in the blog. From what I recall as well, it was a big group of authors on the paper, so it could have kind of been, multidisciplinary in that sense. But that's a really interesting point, isn't it, around the IPCC surveillance.
And the follow-up question. The follow-up question from Lisa is that the infection prevention control team typically wouldn't pick up on Burkholderia as an outbreak organism. It's not an organism that we have on our kind of necessarily on our radar. Very often we specify the groups of organisms that we want to come through from the lab. And it may be that the systems aren't there to pull this one through. So I think identification of these outbreaks could be a bit of an issue, and that may mean we've got an ascertainment bias out there. Perhaps we do have more outbreaks of Burkholderia, to the extent that more than 10% of us would have had experience of managing them potentially.
Yeah, but don't you think that's I kind of understand where the shortcomings might be in the identification, but isn't that kind of where we should be focusing as well for the future for IPC, is how we kind of make sure that we're identifying these organisms that are causing emerging problems or threats, and that if they are causing infections and outbreaks, that we investigate them really thoroughly and routinely, because it's the only way we're going to kind of learn more, isn't it, by investigating these outbreaks.
Yeah, I agree, but it does that approach requires you to have the systems, the surveillance and the data and epi to actually flag the outbreaks in as close to, I'm going to use the words now, as close to real time as you can. Because if you have a system that years later somebody's saying, oh, retrospectively based on, I don't know, an academic study we did in our center, we've identified an outbreak, that's no good at all. You need to be there in the moment to actually understand. Okay, there's a follow-up.
There's some feedback on your poll, John.
Oh, is there? Oh dear. I hope people like it.
Should include all of the above, which is a great comment.
Okay, all right. Let's have a look at these results then. Thanks to those who voted in this second poll. So, what do you think is the most important factor in preventing outbreaks of Burkholderia? Contamination of products, hand hygiene, environmental hygiene, or antimicrobial stewardship? And yeah, thanks, Jeremy. Good point. Should have had all of the above. I'll take that. Hands up. Next time, I'll try and include that. But so most people think contaminated products. I think that's reasonable. Quite a big chunk think hand hygiene. An even bigger chunk think environmental hygiene, and nobody suggests antimicrobial stewardship. What do you think, Phil?
I was all of the above. No, I'm pulling your leg. Prevention of contamination.
You can do the polls next time then, Phil.
Yeah. Prevention of contaminated products was my response. Just because in my experience, that's where I've seen these outbreaks originate from, and kind of where I've experienced reading about them in the literature. What about for you?
Well, I chose preventing contamination of products, but I wonder whether there may be an argument for antimicrobial stewardship, which nobody's voted for. Because if you don't have an environment with a selective pressure for given agents, then the prospect of an outbreak with an organism with that resistance pattern emerging are significantly lower. So maybe even if you do have some deficiencies around product management or hand hygiene or environmental hygiene, if you haven't got that overarching selective pressure of antibiotics, perhaps they don't often translate into outbreaks. Yeah, and I suppose when it comes to prevention, we have to have all of the above, don't we? We can't just choose to focus on one aspect of prevention in the hope that it will deal with the issue. Okay, I wanted to ask you about environmental sampling and the sampling that was done as part of this outbreak. Over what period was the sampling done? Do you know, and how many items did they sample? Was it a huge number or was it a relatively small amount?
I don't know the period, but I think in terms of the number of items or samples that were taken, it wasn't that high. I think it was around about the 50 to 60 mark.
Okay. And did they explain the way that they sampled the syringes, or was it the syringe drivers or the actual individual syringe vials? Did they go into detail about that?
I don't think they actually specified. I think they just talked about the kind of device as a whole.
Okay.
But it would be interesting, wouldn't it? I mean, they talk about identifying limitations in the preparation of some of the IV fluids. So it'd be interesting to kind of understand where in that process, the risk and the contamination was coming from.
I mean, presumably these devices, wherever they sampled them, were not contaminated at source. They weren't coming out of the factory with this antibiotic resistance. I nearly said Pseudomonas then. I know it is related to Pseudomonas. They didn't come out of the factory contaminated with this Burkholderia antibiotic resistant version. So at what point in the chain did these vascular access items become contaminated, do you think?
Must have been in kind of the preparation and handling of the IV fluid. So probably, just before administration. And I'm really interested in how they weren't able to understand where the source was. I think, that's the missing piece from this paper is where the source actually came from.
Yeah.
Staff, I'm scratching my head really. I don't have all the information, but I'm just wondering where it came from.
Yeah, or maybe there wasn't kind of a point source. Perhaps it was a patient developed an infection from outside the hospital, perhaps from another centre transferred in. And this was a relatively simple case of patient-to-patient spread over a period of time, but involving these contamination of these objects. And it may be that they were linked with the positive blood cultures, and perhaps some of them were contaminants and some of them were true infections. It would be really good to know more about the clinical picture for those patients who were infected.
Definitely.
Just to know. Okay, let's talk some more about whole genome sequencing then. In this study, all 19 had the same clone. So was that pretty slam dunk? These are all so closely related.
Yeah.
There's no other way. It must be patient-to-patient spread.
Yeah, that's the kind of beauty of the whole genome sequencing. They were able to get into that real high level of detail and confirm that the similarity between them was so similar that it was, like you say, it was a slam dunk. And I think that actually to be able to do that within a group like the BCC complex, where they are so closely related, and the tiniest bit of variation could mean you're into a different species. To be able to identify that similarity at that level just shows the power of whole genome sequencing.
Okay. So this is probably going to be quite hard to answer, but I'm going to ask it anyway. If they hadn't used whole genome sequencing, how would that have changed the management of this outbreak?
I wonder whether or not they would have detected Burkholderia as being the kind of principal outbreak organism in the first case, because they'd clearly, linked it back to originally thinking it was the blood culture contaminants.
Yeah.
I guess as time goes on and it keeps cropping up, then you've got to think about something else.
But there's an eventuality where they might have said they're all contaminants and never kind of considered them part of an outbreak. Interesting. Yeah.
Yeah. I mean, the other thing as well, we don't have all the information, but they did talk about co-infection with other organisms and being treated for other infections from, they don't list the organisms, but I just wonder if they would have just thought that these patients have got infections, and they aren't linked, and they are then being treated that way. I think though, if you're seeing these organisms crop up over a prolonged period of time, there have to be some investigation. And I think my first port of call would have been contaminated products, knowing what we know from the literature, and the evidence there is about these organisms being able to persist and thrive in lots of things like ultrasound and wipes and chlorhexidine mouthwash, etc. So if they hadn't had the whole genome sequencing, I probably would have gone there in terms of trying to identify the source in the environment or from the products or consumables that were being used.
Yeah, curious though that they didn't identify it in any of the products they sampled, because they sampled, by the looks of it, pretty much everything they could lay their hands on. I think maybe that contaminated product that was part of it, that was the root cause of the outbreak.
Yeah, yeah. I mean, I'd love to see a follow-up paper to identify where the source was. Just missing that one kind of vital bit of information.
But yeah, it's pretty uncommon to find a smoking gun source of an outbreak. It's so often
Yeah.
like this. You've got, oh, well, it could be this, might be that. Found this contaminated, but was it cause or effect? It's often difficult to know the direction of travel, even when you do find an item that's contaminated.
Yeah, I agree. I guess maybe then it would be something around the follow-up, because they did identify poor practice in the IV management. So did actually changes that well, did it stop the outbreak is the other key question. And then they might have had a bit more of an inkling as to whether or not that was the source, and whether or not they could get on top of the outbreak by making changes that way.
So what exactly did they change? Did they take any action about these contaminated syringes? Did they enhance disinfection? You mentioned they'd made some educational interventions around vascular access.
They don't stay. So it's just quite vague, actually, in terms of the limitations that they identified, and they don't actually go into detail about the limitations. I think they focus more the paper on the whole genome sequencing side of things as part of the investigation of the outbreak, rather more around the IPC interventions. I mean, one of the reasons I put for picking this paper out was I was really interested to see how they got on top of the outbreak and what IPC measures they put in place.
Yeah,
But they don't really call that out, and they
but that
don't
wasn't, yeah,
identify the source of the outbreak either. So there's a couple of questions left to be answered.
for sure. Okay. So you mentioned that in terms of the whole genome sequencing aspect of this piece of work, there was a need for a multi-professional collaboration. I wonder if you could just expand on who was around the table for this outbreak involving the whole genome sequencing, and whether that's different to the normal crew that we might consider part of an outbreak committee.
It's a good question. So IPC, obviously key components of that outbreak management, microbiologists, and then they talked about a molecular biologist presence, and I'm assuming they probably mean a bioinformatician in that as well.
Yeah.
So this is all down to kind of the interpretation of the data that the whole genome sequencing is spitting out, and being able to look at, you know, the similarities in the data that you're looking at between these organisms. So it is a different group of people around the table for identification when you're using these different techniques.
Yeah, and I suppose it's that bioinformatician slash molecular biology expertise that we're not so used to having as part of the team.
Agree. Yeah, I mean, it isn't something that we normally have within our outbreak management, and I think that's where the future needs to go, I guess, if this is going to be implemented in other healthcare organisations and be effective. It's how do we work with, you know, the data and the information that is being provided by using these different techniques, and then kind of bundled up into a way that we can make informed decisions around outbreak management from an IPC perspective.
Yeah, I agree. I think there's some potential unintended consequences if we don't get it quite right, because we could end up with huge volumes of genomic information around all kinds of organisms, but actually without some clarity of interpretation, we could end up even more confused than we started. I've certainly been involved in outbreaks, and the typing comes back, and you're like, well, I'm none the wiser. That's actually made it more confusing and not less confusing, because of the way that the interrelatedness between isolates is not fully understood. I think
Mm-hmm.
it's more of a bioinformatic gap rather than a purely genomic gap.
Yeah, no, I agree. And I think that's where the future needs to be addressed. There is one more question in the chat, John.
Oh yeah, okay.
Maybe I'll ask you,
Yeah, go on then.
would you consider BCC, so Burkholderia cenocepacia complex, as a contaminant in practice?
Not usually, no. I think it would usually be the gram-positive skin-related organisms, your coagnex staphs, that you would be putting down to contamination. So I think it would be a stretch to throw it out as a contaminant. But if you had a little cluster of them, yeah, you might begin to wonder about vascular access line care, as they did. And I think the finding of the contaminated syringe does make me a bit suspicious that this could have been an issue of an outbreak of bad practice as much of an outbreak of an organism.
Agree. Good answer.
Okay, so we are pretty much out of time. Thank you, Phil, for bringing that to us. It was really good to hear more about Burkholderia, not an organism we talk about so often, and hear how whole genome sequence really helped with the resolution of the outbreak. Next up, in a few weeks' time, we have IPC Partners Journal Club in partnership with the Healthcare Infections Society. And we'll be looking with Siobhan Kelly, along with Phil and I, at an MRSA article collection, and that's across both of his journals, the Journal of Hospital Infection and Infection Prevention in Practice. So we'll be taking an article each from that special issue. And then on the 18th of June, we have an IPC Partners Journal Club in partnership with Monica to look at risk factors for post-op SSI with Staph aureus. So please do take a look at the website. It's got the schedule for upcoming journal clubs. So we try and keep it up to date as things change. Previous recordings from journal clubs are available on the YouTube channel. And thank you, as ever, to IPC Partners for hosting today. And thanks for Phil for leading us through our paper.